Rahim. Let's discuss the anatomy of the ventricular system of the brain. And this chart over here, the diagram shows uh, a kind of a lateral view or phantomized view of the ventricles of the brain and their interconnecting channels. And note that all the parts of the brain, all the 5 secondary brain vesicles, including the spinal cord, they contain a remnant of the neural canal, which because everything starts from a hollow neural tube, and the neural tube is engulfing a neural canal. So every part of the brain that develops from the neural tube encloses a canal, and which is expanded later to form the ventricles. So, and it contains a remnant of the neural canal, and it expanded in some places to form the ventricle. So now let's discuss the anatomy of the ventricles. Both the cerebral hemispheres in the telencephalic location, they contain the lateral ventricles. This is since this is the lateral views, only one is shown, but remember that there are 2 of them. This is the lateral ventricle. This entire ventricle is the lateral ventricle, and it has parts that encompass all the lobes of the cerebral hemispheres. So the anterior horn of the lateral ventricle is in the frontal lobe. The body, this is the body of the lateral ventricle is present in the parietal lobe. His posterior horn is projecting into the occipital lobe, and this inferior horn of the lateral ventricle is extending into the temporal lobe. So, but just remember that there are 2 lateral ventricles. There are 2 of these, each one having a anterior horn, a body, a posterior horn, and an inferior horn in both the cerebral hemispheres. Now, both the lateral ventricles, they communicate with the third ventricle. This is the third ventricle, and it is enclosed on both sides by the thalamus, the diencephalon, and the lateral ventricles, they empty into the third ventricle through the foramen of Monro. Both the lateral ventricles, they are they're 2 foramina of Monro, each one from each lateral ventricle entering into the third ventricle. And then the third ventricle communicates with the fourth ventricle. This is the fourth ventricle through the cerebral aqueduct. This cerebral aqueduct is going through the midbrain, and this is the only part of the brain that does not contain a ventricle. And so the cerebral aqueduct is not the ventricle. It's not a ventricle. It's a channel that is connecting the third to the fourth ventricle. And then the fourth ventricle then is in communication with the central canal of the spinal cord in the upper medulla. The fourth ventricle is in the upper medulla, and it communicates with the central canal of the spinal cord. And the next point is that in parts of all of the ventricles, we have the choroid plexus. In all the 3 ventricles, the lateral ventricle, the third ventricle, there is a single third ventricle, 2 lateral ventricles, single third ventricle, excuse me, and the fourth ventricle. They all contain choroid plexus. Now, choroid plexus is a specialized form of epithelium, and which contain choroidal arteries. And the choroidal arteries, through the choroidal arteries, the CSF is produced, and it is just an ultrafiltrate of the plasma. So the choroidal arteries will produce the CSF in these ventricles, in these locations. It is present in the lateral ventricle, the third ventricle, and the fourth ventricle. So the choroid plexus is present in the body, mainly located in the body and the inferior horn of the lateral ventricle. It is present in the lateral wall of the third ventricle, and it is present somewhat in the roof of the fourth ventricle. These are the main locations of the choroid plexus: the body and the inferior horn of the lateral ventricles, the lateral wall of the third ventricle, and the roof of the fourth ventricle. Now the next point is that the amount of CSF produced in a particular average human being is about 400 to 500 cc's every day, but the entire volume of the ventricular system plus the subarachnoid space, which is shown in blue over here, the entire volume, which is the subarachnoid space, is these are the blue part is the dural venous sinuses. I'm sorry about that. The subarachnoid space is this space. So the entire subarachnoid space and the ventricular system, the capacity of that is 150 cc's. So the point being that CSF is a circulated fluid which has to turn about 3 to 4 times over in a day, and it has to be secreted by the choroid plexus, and then it has to find an exit point out into the subarachnoid space in order to avoid compression of the brain because of the pressure that it will exert, because the production is more and the space that it can occupy is less. So it has to find an exit point from the ventricles into the subarachnoid space, and there are only 3 points present in the fourth ventricle. It is the two foramina of Luschka, which are present in the lateral part of the fourth ventricle, and the CSF can get out from there into the subarachnoid space, and one foramen of Mijandi, which is shown right over this foramen of Mijandi, and these are the two. There are two of those foramina of Luschka, and it's at these points that the CSF can get out from the ventricles into the subarachnoid space. So as you can see, the arrows are indicating the direction of flow of the cerebrospinal fluid, that it has to circulate both into the ventricles as well as the subarachnoid space. So it is produced inside the ventricles. This is the third ventricle, but it is also produced in the lateral ventricles. The CSF from the lateral ventricles, both lateral ventricles, flows through the foramina of Monroe into the third ventricle, and from the third ventricle it has to pass through the narrow cerebral aqueduct into the fourth ventricle, and from the fourth ventricle it exits from the 3 openings, the 2 foramina of Luschka and 1 foramen of Mijandi, into the subarachnoid space, and from the subarachnoid space it circulates all over outside it circulates in the space outside subarachnoid space, outside the brain and the spinal cord, all the way to the tip of the spinal cord, and then this subarachnoid space, it protrudes these small knobs which are present over here. You can see these knobs are protruding into the superior sagittal sinus, and then it exits from the superior through these arachnoid granulations into the dural venous sinuses, the superior sagittal sinus. So these arachnoid granulations are everywhere in the subarachnoid space, which protrude into the dural venous sinuses. So it's starts from the ventricles, produced by the choroidal arteries, gets out from the lateral ventricles to the third ventricle, then the cerebral aqueduct into the fourth ventricle, from fourth ventricle into the subarachnoid space, and from the subarachnoid space, it through the arachnoid granulations, it goes into the dural venous sinuses. So this diagram is a coronal section of two of the dural venous sinuses, and one of the most important of those two is the superior sagittal sinus. The superior sagittal sinus, and as far as the drainage of the CSF is concerned. So as you can see, it's a coronal section, and you can see this is the yellow part is the subarachnoid space in which the CSF is circulating, and these arachnoid granulations, they are protruding into the dural venous sinuses, and CSF flows from the subarachnoid space into these arachnoid granulations and into the dural venous sinuses, and it follows its concentration gradient, because there's greater pressure inside the subarachnoid space, and there's low pressure inside the dural venous sinuses. So because the hydrostatic pressure of the CSF, it flows from the subarachnoid space into the dural venous sinuses. Let's discuss some of the forms of hydrocephalus that occurs due to the obstruction of the CSF flow. And basically it's of two types: hydrocephalus, non-communicating and communicating type. The non-communicating type of hydrocephalus occurs when there is obstruction of flow within the ventricles. At some point in the ventricles, there is obstruction of the flow of CSF within the ventricles. Most commonly, it occurs at narrow points. Most popular points are the foramen of Monroe and the cerebral aqueduct. So any obstruction inside the ventricles, or the communicating channels, the interconnecting channels which are connecting the ventricles, if they are obstructed, then this is known as non-communicating hydrocephalus. The communicating hydrocephalus is not an obstructive problem. And by the way, non-communicating hydrocephalus is also known as obstructive hydrocephalus. And the communicating hydrocephalus is a reabsorption problem. If there is some problem in the arachnoid granulations, for example, in some forms of meningitis, arachnoid granulations, they become inflamed, and that obstructs the flow of CSF from the subarachnoid space into the dural venous sinuses. So this is a communicating type of hydrocephalus. And then any tumor within the subarachnoid space could cause communicating hydrocephalus. Any tumor within the subarachnoid space, or it could be a choroid plexus tumor on rare occasions, in which there is overproduction of cerebrospinal fluid. This is also known as an ependymoma, which produces excess CSF. So this can cause communicating hydrocephalus. Another form of hydrocephalus is normal pressure hydrocephalus, and this is a form of hydrocephalus which is just like a communicating hydrocephalus, because it's an absorption problem in the elderly. It happens in the elderly. There is neuronal loss, there is atrophy of the brain, so the ventricles are chronically dilated. But that is in contrast to the communicating hydrocephalus. The pressure of CSF is normal because of the atrophy of the brain, and the brain substance is low. The brain substance has shrunk, and the ventricles are chronically dilated, so it compensates for the pressure. Pressure is normal. In communicating hydrocephalus, the pressure will be elevated. In normal pressure hydrocephalus, the pressure will be normal, but it is pretty much the same condition as communicating hydrocephalus. So the ventricles are chronically dilated and produces a triad of signs and symptoms, which is dementia, an apraxic magnetic gait, They tend to walk with their feet, you know, as if their feet are stuck to the ground. That is known as a magnetic gait. And there is urinary incontinence. And the solution is a peritoneal shunt. The urinary incontinence is because the bladder control neurons, which control the bladder, they are present near the some part of the lateral ventricle. So that causes the urinary incontinence in normal pressure hydrocephalus. So three major characteristic signs and symptoms are dementia, apraxic gait, because they shuffle their feet on the floor as if their feet are stuck to the ground. They cannot raise their feet as well as the other normal people can. And then the last one is the hydrocephalus ex vacuo. It occurs because of excess CSF in regions where brain tissue is lost due to atrophy, example Alzheimer's dementia, or due to stroke if there is some portion of the brain that becomes atrophied due to the stroke, or surgery, and there is some loss of neuronal tissue. It causes an excess CSF in these regions, and the ventricles are dilated, but the CSF pressure is normal. So in contrast to these people, uh, uh, there is some kind of neuronal loss in a specific area or region of the brain. Instead, this occurs in when there is overall atrophy of the brain. This occurs ex vacuo when there is some portion of the brain that is missing due to stroke or surgery. So that is known as hydrocephalus ex vacuo.